So this is a super cool plant, but before I talk about Polygala, I want to sort of explain the unique geologic history of Florida that has contributed to what is the central ridges of Florida, which harbor a very endemic flora. So during the Pleistocene, Florida and the rest of the Earth underwent 11 major glacial events. During these times, as ice levels would decrease, you see that the Florida coastline would be inundated with water, and you would have just what they call the central ridges, or the islands, or the ancient islands of Florida above water. So these acted as a refugia for plant and animal species that were able to migrate up onto the ridges to avoid the water. And as such, they sort of have adapted to this unique environment, which is said to be sculpted by the sea and maintained by fire. They mean the sculpted by the sea, and this is one of my favorite quotes, but what they mean by that is that as sea level rise, it would push sand and soil to the center part of Florida to help build up the ridge, and then maintained by fire is alluding to the fact that there's many frequent wildfires in the area that plants and animals are adapted to. So currently there's about 26 federally endangered plant and animal species that occupy the ridge. So one part of the ridge, concentrated in Highlands and Polk County, is the Lake Wales Ridge. Here we can see a picture of what was pre-settlement Florida and an estimation of the intact habitat at that time. And you can see that in 2003, there's been a large decrease in the amount of intact habitat. So currently there's only about 15% left. So it's sort of sad that this area that harbors this unique flora and fauna is being destroyed at a rapid rate. So this fragmentation has cut off many of the species that were once continuous across this ridge, as well as the ridges more north in Florida. So things that have caused destruction of the ridge are things like residential expansion, as well as fire suppression to protect residential houses, agricultural investment, citrus groves are very prevalent in this area with the type of soil, as well as all-terrain vehicle traffic. So these have all contributed to the fragmentation and deterioration of this ridge habitat. So the plant that I worked on is Polygala lewtonii. It's federally endangered. It's a perennial herb. It forms multiple stems and gives it this nice little bushy appearance. And it has a very long taproot to tap into water, because this ridge is very dry at most times. It's found only in the yellow sandy soils of the ridges of Florida, so that includes the Lake Wales Ridge, as well as the ridge to the north, which is the Mount Dora Ridge. And it's adapted to frequent burnings of its habitat. So after a fire, you'll see many Polygala seedlings sprout up all over the place, many more than what were there before. So here's sort of the habitat that Polygala would occupy. So it ekes out its existence in any little pocket that it can find in this habitat. Right now you can see it looks pretty densely packed, but after a fire, you would see many of these saw palmettos and lichens on the ground be completely wiped out, and Polygala would be able to recolonize this area. So what's really interesting about Polygala lewtonii is it has 3 different types of flowers. It has above ground, open, cross-pollinated flowers. These are termed chasmogamous. You can see they occur on the terminal raceme, and you can see that it forms multiple flowers on that. Then it has above ground closed self-pollinated flowers. These occur in the leaf axils, and these are termed cleistogamous. You can see we have a picture of some of the fruit that are produced by this flower, as well as the really interesting flower, which is the below ground closed self-pollinated flower. So this is producing a flower along the rhizome that is producing pollen and being self-fertilized below ground. So it's not a clonal type of mechanism. This is truly a flower below ground, which I think is really cool. So here we can see one Polygala lewtonii out in the wild, and we can see one of these flowers marked by this red arrow. And you can see that these rhizomes, you can see that they sort of can spread quite a distance away from the maternal plant. So the other really unique thing about Polygala lewtonii is it produces 2 different types of seeds for different ecological roles. It has its above ground seeds, which are able to be dispersed by ants. You can see this little yellow elaiosome. This is attracted, ants are attracted to this, so they'll bring it back to their nest, remove it, and it's found that that has no effect on the germination. So those are able to be dispersed, whereas the below ground seeds are thought by, or hypothesized by other researchers like Cheplik, to be a reproductive strategy to sort of recolonize your maternal site after disturbance. So as far as Polygala is concerned, there's many wildfires in the area, so it's nice to be able to recolonize the site as quickly as possible. And these seeds are slightly bigger than the above ground seeds. These ones are a little bit immature, but, or these ones are the above ground seeds, but they're slightly bigger and a little bit more well provisioned, so they can come up a little bit quicker. So here is the problem that we see when we find Polygala out in the wild, at least for a conservation person trying to protect the species. We see one maternal plant surrounded by all these seedlings, and we really have no clue how these are being produced. Were they being produced by above ground flowers or below? Were they being produced by self-fertilization or outcrossing? And this phenomenon has sort of big ramifications for how you would preserve the species. Are each of these individuals contributing to the genetic diversity of the species, or are each of them self-individuals such that they be very similar? So you would want to protect the population as a whole, and maybe not every individual. So in order to tackle this problem, we asked a couple research questions. We wanted to know what was the primary mode of reproduction in Polygala lewtonii, whether it be self-fertilization or outcrossing, considering it has a mixed system. And then we wanted to know how that was affecting the patterns of genetic variation across the landscape. So in order to look at this, we did a stratified sampling scheme. We first started on a fine scale of about 1 kilometer squared at the Carter Creek South. And we looked at 4 blocks of 72 individuals each. The blocks are grouped by each of the like symbols are one block together. And those are each subdivided into plots, so that is one symbol. And we wanted to do this to try to understand the distances of sort of seed dispersal and pollination. So the idea of the plots, or the small circles, is to try to understand the distance that seeds might spread along the rhizome. Whereas within a block, we figured that was about the distance that ants might be able to disperse seeds. In between blocks, we figured that was the distance that pollinators could easily traverse. And then we also looked across the range-wide scale of the species at 3 other protected sites, Lake Wales Ridge, Scrub Point Reserve, and Ocala National Forest, just to get an idea of the patterns across the full range of the species. And in total, we collected 360 tissue samples. So for the analysis, we developed a set of 11 microsatellite markers to genotype these individuals. We used some common genetic diversity metrics, as well as looked at patterns of genetic structure with a pairwise distance metric, as well as Bayesian clustering programs. So what we found is that the observed heterozygosity was always much lower than the expected heterozygosity, and that the observed heterozygosity was very low in the species. So this is telling us that there's some amount of non-random mating going on in these populations. In looking at an inbreeding coefficient, we find that it's also very high for all the populations, even the ones on the very fine scale. So looking at some of the genetic structure of the species, this is a pairwise genetic distance tree. So the branches are scaled to the amount of genetic distance between any population. And you can see that what's really interesting to us is that each of these populations comes off on a very large branch. So some of these populations in this area are the distance of 10 to 20 meters apart, but we're seeing that they're very genetically distinct from each other. So it's sort of something that was very unexpected for us. Usually when we see these, when anyone within one of these blocks usually forms very small branches, very genetically close for the amount of geographic distance we see. So this is already showing us that there's very little gene flow occurring between most of the populations, and that there must be very little outcrossing occurring within the species. So on to the structure results, we found that there was 9 optimal clusters, and each cluster in this graph is going to be represented by a unique color. Each individual is represented by 1 line, and we see that each individual can vary in the amount of proportion assigned to any one cluster. So on the range-wide scale, we find that they each form basically one distinct cluster. We see between Lake Wales Ridge and Ocala National Forest, there are what looks to be some individuals that were dispersed into this area, but it's sort of, we're really unsure as to how this would have come about, considering that ants are not able to traverse the area. The distance in this area is something of like thousands of kilometers. So we're sort of baffled by this. Maybe it's some kind of long distance dispersal event. but we really don't have a good answer for this. On the fine scale, where the patterns are really interesting, we see 6 distinct genetic clusters within a kilometer squared area. So we see that there's just about 1 cluster each in the Carter Creek South 1 and Carter Creek South 2, whereas the Carter Creek South 3 and 4, we see 2 distinct genetic clusters. So looking at the self-fertilization rate estimated by Instruct, we find that taking into account the genetic clusters that we found, about 87% of the reproduction of the species is through self-fertilization. And we see that that is the same, whether it be on the fine scale or on the branch-wide scale. So mapping that back to the map that I showed before, you can see that the blocks are going to be represented by the colors from the structure graphs. And what you see is that there's very little gene flow occurring between any of these pie charts. The only place where we really do see gene flow in individuals being dispersed is on an extremely fine scale. So between Carter Creek South 2 block and Carter Creek South 3. So there's some amount of individuals that are being outcrossed and being produced, and some amount of seeds being dispersed into different areas, but it's still on a very fine scale. We're not seeing it the distances that we would have expected, considering that it produces this nice raceme of very open outcrossing flowers. So this led us to conclude that the primary mode of reproduction is via self-fertilization. There is some confounding issue with inbreeding, that if the individuals are produced by selfing, and then you have pollination occurring between 2 selfed individuals, that's sort of going to still have the appearance of being self-fertilized. So that's sort of an issue that we're looking into with some other experiments at Archibald Biological Station, doing some pollination and bag work. And then we see that there is a small amount of outcrossing, but it's only very limited. And that this has had a pronounced effect on the structure of genetic variation. We see that the populations are very differentiated, even at a small scale, and that pollen and seed dispersal are only occurring with populations that are on very geographically close distances, something like 10 to 20 kilometers, or 10 to 20 meters, sorry. So this sort of answers our question here. Most of these individuals that you see around here are going to be produced by self-fertilization. So as far as conservation, we know that habitat loss is still going to remain the biggest threat to Polygala lewtonii. And we know that we need to protect the full range of genetic diversity, and to do that, we need to protect every population. So we figure as far as buying land is usually very hard for these conservation agencies. A good conservation intermediate effort would be to retool the seed banking efforts to incorporate the scale that we worked on and collect seeds from individuals at 10 to 20 meter distances in order to protect the full range of genetic diversity at any one site. And we would like it if it could begin with unprotected sites, just to sort of seed bank those for the future in case we can't procure those lands. So with that, I'd like to first thank David Vogler. So he was the REU coordinator for the mobile REU when I did this project. And then all the work with field assistants, all the people who provided me nice photos, especially Richard Abbott, who provided a large amount of photos of Polygala, all different kinds, of all different areas in Florida, as well as all the funding agencies that made this work possible. Thank you.
We have time for a question?
Yes.
So you mentioned that you've got rhizomes there. Have you seen any indication of actual vegetative growth, or you've got new plants coming out from the rhizomes?
We don't see any of that. So the one thing that sort of makes that kind of observation difficult is it's federally endangered, so digging at the roots is sort of a difficult proposition. But a lot of the work of Carl Weekly on this plant has been to try to eliminate that hypothesis, and they found no evidence of it, even with all the work they've done. So it really is producing a flower underground, which is, in my research, at least what I did for the plant, very rare across all plant species.